In this section, we are going to continue our discussion of transcription, or how the DNA code is made into an mRNA. Here are our lecture objectives for this part of the chapter. As always, I expect you to have these mastered before our exam. Please let me know if you are struggling with any of these objectives. In this part of the lecture, we will continue covering the process of transcription, or making an RNA copy of a DNA gene. At the end of part 2, we will start on the topic of translation, or going from an RNA copy of a gene to the actual protein. Now that the RNA polymerase is bound, the process of transcription can begin. In eukaryotes, there are 3 types of RNA polymerase: 1, 2, and 3. These polymerases all transcribe specific types of DNA. 1 and 3 transcribe the RNA that will not be translated into proteins. The 2 main examples are rRNA and tRNA. RNA polymerase II is responsible for transcribing the genes that will eventually be made into protein products. As we talk about the process of translation or transcription, if I am not differentiating the polymerases, I will be referring to RNA polymerase II. The process of RNA polymerization is very similar to that of replication, with the differences we noted earlier. The RNA polymerase will move along the DNA sequence until it reaches the terminator sequences. These sequences are at the end of the gene and tell the polymerase it can be removed from the DNA and the mRNA released for processing. In the eukaryote, the RNA polymerase is phosphorylated to provide the functionality of the enzyme. When the terminator sequence is reached, the enzyme becomes dephosphorylated and is removed from the sequence. Overall, this process has fewer components than replication and can seem very simple. While on the surface that is true, I in the textbook have covered just an overview of both replication and transcription. For this class, I don't expect you to know much more than the basics we have discussed here, but be aware that there is more to the story. In eukaryotes, the process is not completed with the making of the transcript. The transcript must be processed in order to stabilize the transcript and to remove unneeded parts. The transcript is stabilized by the addition of the 5' cap and the poly A tail. Unneeded parts are removed during RNA splicing. The first level of processing actually occurs while the transcript is being made. In eukaryotes, the mRNA is located in the nucleus, while the machinery for translation is in the cytoplasm. In order to get the mRNA out of the nucleus, the sequence must be processed. This processing is much like being issued a passport. In order for the cell to keep the DNA in the nucleus and allow the mRNA to pass through the pores, there must be signals added to the mRNA molecule. There are 2 specific modifications made to the mRNA molecule. The first of these is the 5' cap. This cap consists of a guanine with a methyl group attached. This cap is added to the 5' end of the mRNA. The other addition is the poly A tail to the 3' end. Just as the name implies, a series of adenines is added to the 3' end of the mRNA transcript. With these additions, the mRNA can be exported from the nucleus. Some research has indicated that this process also stabilizes the molecule and helps the translation enzymes recognize it as an mRNA transcript. The transcript is almost ready for exportation, but before it can be sent out for translation, the introns must be removed. What are these introns, you ask? In the eukaryotic mRNA transcript, there are 2 regions, introns and exons. Exons are the regions that actually code for the gene product, while introns are non-coding regions of the transcript that must be removed. To remove these introns, the transcript must undergo splicing. To begin the process of splicing, the introns must be identified through a specific sequence within their nucleotides. The spliceosome then will help the introns form a lasso-type structure. This lasso is then removed and the exons are sealed together for a complete transcript. This spliceosome consists mainly of RNA components known as small nuclear RNAs, or SNRPs for short. One of the benefits to this process is that genes can be spliced differently by the spliceosome. This creates a variety of proteins that can be coded for by one gene. Now that we have processed the mRNA completely, it is ready for exportation from the nucleus. This is when the modifications we discussed earlier come into play. Without the cap and the tail, the mRNA would not be able to pass through the checkpoints of the nucleus. These checkpoints are nuclear pore complexes. These complexes check for the cap and the tail of the mature mRNAs and do not let the introns or partially transcribed mRNAs into the cytoplasm. This mechanism is extremely important because it keeps sequences of mRNA that are not meant to be expressed from the machinery that could create proteins out of them. This is different from what happens in prokaryotes. In prokaryotes, the cell may start translating the mRNA transcript before the process of transcription is complete. Since prokaryotic genes do not contain introns, this is a good way to express genes quickly. Mature mRNAs have a sort of expiration date, so to speak. Once they have been used to make the required number of protein copies, they need to be destroyed. Just leaving these bits of transcripts floating around the cell would be disastrous, as the longer a mature mRNA strand stays in the cytoplasm, the more proteins that are made from it. That has the potential for exceeding the energy capabilities of the cell, or could result in an injurious excess of protein. And within the nucleus, we need to get rid of the introns. Both mature mRNAs and excised introns are degraded over time by enzymes called RNases. The length of time mature mRNAs are allowed to persist may be determined by a sequence within the tail region of the mRNA. Each mRNA transcript has a specific length of time before it starts to be degraded. Some of these can be more than 10 hours, while others are less than 30 minutes. The moral of the story is that RNases aid the cell in cleaning up used transcripts before too much protein is generated. As we wrap up transcription, it's time to explore everyone's favorite connections moment, evolution! As you can tell from the differences we have described between prokaryotic and eukaryotic transcription, a lot has transpired over the millennia, but the heart of the process has remained unchanged. All cells use RNA polymerase to create an mRNA transcript. The differences in the processes can help us see the divergence in these cells. There's a debate going on about the presence of introns. Some believe that all cells used to contain them, and the prokaryotes simply decided to remove them for quicker transcription translation. Other researchers believe that these regions are actually left over from mobile genetic elements acquired from intracellular parasites. Regardless of what the final conclusion is, the differences in these absolutely universal processes can add one more part to the story of evolution. We have finally made our transcript. That means that we are officially halfway done in this process of generating proteins. Now we get to talk about the process that takes us from RNA to proteins, otherwise known as translation. In this section, we are going to discuss the nucleotide coding system, revisit our friends tRNA and rRNA, review the ribosomes and their role, and lastly, talk about the process of creating proteins as well as how we destroy these proteins when they have reached the end of their use. I am sure that at some point in your scientific experience, you have had to take an RNA code and create an amino acid sequence from it using a specific chart that shows you how the two come together. In these kinds of charts, like the one you see here, the first two nucleotides are very important in determining the amino acid that should be added to the sequence, while the last nucleotide does not particularly matter. This last nucleotide is sometimes referred to as the wobble base, meaning it can be interchanged without changing which amino acid is coded for. These 3 nucleotides, or triplets, form what is known as a codon. These codons are always read within the same reading frame or window. If an aberrant nucleotide were to be added to the sequence, this could result in an entire frame shift where 2 nucleotides of one codon are read with 1 nucleotide of the next codon. This can change the entire structure of the protein. How are these codons read? This is where transfer RNAs, or tRNAs, come into play. As you can see from the images on this slide, the tRNAs fold in to form a 3D structure. tRNA can be represented in numerous ways, but there are 2 important regions on each of these molecules. The first is the anticodon region. This is the region that is complementary to the codon sequence found on the mRNA. Using the anticodon, the correct tRNA will always bind to the mRNA, and the correct amino acid will be bound to the protein being built. At the opposite end of the tRNA is where the amino acid is attached. Each tRNA codes for only 1 amino acid, giving it the needed specificity. These tRNAs dock within the ribosome to transfer the amino acids they carry to the final protein product. Well, that's it for this part of the chapter. In the final lecture, we will continue the topic of translation.